Hello, I'm Kate Corrigan, Dean of the Graduate College here at UNLV. Welcome to our Inspiration, Innovation, and Impact celebration of graduate student research. I'd like to introduce you now to Jacqueline Fan from the Department of Chemistry and Biochemistry. Jacqueline studies with Dr. Ernesto Abel Santos, and the title of her presentation today is Preventing a Foul Epidemic, Prophylaxis Against Clostridioides difficile Infection, or CDI. So Jacqueline is a doctoral student in chemistry. She studies Clostridium difficile infection, or C. diff as it's called, a hospital-associated disease that impacts mostly the elderly and immunocompromised. With increasing hospitalizations due to the COVID-19 pandemic, the likelihood for also acquiring CDI is concerning and dangerous. Jacqueline believes that informing the public about precautions and scientific advancements towards combating infectious diseases is more critical now than ever. Let's hear from Jacqueline. Jacqueline.
Hi, my name is Jacqueline Fan, and I'm a PhD student studying under the advisement of Dr. Ernesto Abel Santos in the Department of Chemistry and Biochemistry. Now, let me first begin by taking you back in history just a few years ago when I presented at the UNLV 3-minute thesis competition. In my 3-minute thesis talk, I mentioned how microorganisms would contribute to the greatest and most urgent global risk. Now, here we are, several years later, in the midst of a pandemic that was caused by a virus that has now claimed over 500,000 lives in just the United States alone. COVID-19 might be the most notorious pathogen of our current times, but other infectious diseases have actually been around for many years, wreaking havoc on our communities. One of these diseases is Clostridioides difficile infection, or CDI, which causes a very foul-smelling epidemic. CDI is caused by rod-shaped bacteria called Clostridioides difficile, or C. diff. A key characteristic of C. diff is that it is able to form these tough and round structures called spores that act as the infectious form of the disease. Spores are basically like super eggs that can survive in many different environments until a trigger causes them to hatch, or what we call germinate, into toxin-producing cells that can then eat away at the intestines. This germination process is required for CDI symptoms to occur, and it just so happens that the gut can be a perfect place for these spores to germinate in once the right conditions are met. CDI can cause abdominal pain, nausea and vomiting, explosive diarrhea, gastrointestinal bleeding, and in worst-case scenarios, even death. People who are elderly or immunocompromised, or those who have recently taken antibiotics, are much more susceptible to contracting CDI. CDI is also a hospital-associated infection, so with increased COVID-19 hospitalizations, the potential risk for contracting CDI is quite concerning. In the past year, we have seen all the damage that COVID-19 can cause to individuals, as well as their families, and even to healthcare. These issues are also concerning for CDI. Here are 3 reasons why we should care about CDI too. CDI is a very serious problem. There are over 200,000 diagnoses per year in just the United States alone, with up to 25% reoccurrence within just 2 months of diagnosis. The second reason is that CDI is a very deadly problem. There are over 12,800 deaths reported per year within just 30 days of diagnosis. The last reason is that CDI is a very expensive problem. It can cost an average of $35,000 to treat a single inpatient CDI case, which can then add up to approximately $4 billion in annual cost burden to the United States healthcare system. In this past year, we have been anxiously awaiting the arrival of the COVID-19 vaccine to not only prevent severe cases of COVID, but also to prevent the spread of COVID. Our research also has the same goals, but for CDI. Our research focuses on how bile salts that are produced in the liver and secreted into the intestines have potential to act as prophylactic drugs in the preventative treatment of CDI. In our bodies, there are some bile salts that act as natural germinants of C. diff spores, while others act as natural inhibitors of spore germination. In patients who are immunocompromised or who have recently taken antibiotics, the natural gut bacteria that normally regulate these bile salts are depleted, making them much less efficient at balancing these bile salts. Our lab aims to produce synthetic versions of these bile salts that can act as natural inhibitors of spore germination. After screening hundreds of synthetic bile salts, we have found a compound called kappa that was able to prevent spore germination of multiple strains of C. diff. Kappa was also able to prevent CDI in mice and hamsters as well. Without kappa treatment, infected mice develop moderate to severe CDI, while kappa-treated mice are completely protected from CDI. In hamsters, kappa-treated animals have a much higher survival rate than untreated animals. This shows how much potential bile salts have as prophylactics, and also goes to show how important it is to contribute to scientific research on preventing infectious diseases. During this past summer, our lab temporarily switched gears to join in on the coronavirus testing efforts, converting our infectious disease-focused lab into a coronavirus test-making factory. With lots of careful protocols in place, meticulous attention to detail, and approximately 1,000 volunteer hours, we created over 50,000 vials of solution that were used to efficiently transport specimens to testing laboratories. Like these efforts towards COVID-19, our continued endeavors towards preventing CDI will help make advancements towards reducing the spread of this foul-smelling epidemic. Both COVID-19 and CDI disproportionately affect certain groups of individuals more than others. In fact, we recently found that a high-protein Atkins-like diet actually exacerbates CDI in mice. Therefore, it is important not only to study preventative treatments, but also factors that contribute to increased disease severity. That way, we can aim to protect all individuals from all diverse backgrounds, including those who are most vulnerable. We hope that our research will continue to inspire fresh innovation to help protect our communities and make a positive, lasting impact on our fellow humankind. Thank you and stay safe.